Thanks everybody for tuning in today. As Brittany said last week, I'll be filling in for her for the next couple of weeks leading this seminar, at least hosting. Today, we have Dr. Tyler Mackey. I'm really excited to introduce him. He's an assistant professor at the University of New Mexico. He just started last fall. He's a geobiologist, and his research mainly focuses on studying modern environments to better understand what sedimentological and geochemical evidence microbial communities may leave behind and how to recognize this evidence in the ancient rock record, among other things. He received a bachelor's in geology from Carleton College in Minnesota. He then went to the University of California at Davis, where he got both his master's and PhD working with Professor Don Sumner, where he studied these really cool microbial mats that occur in ice-covered lakes and in Antarctica. And I think he's going to be talking a little bit about that today. And this is actually where I met Tyler, as we had the same PhD advisor, and part of my PhD was studying the sedimentology of these same lakes that he was studying. And so we've known each other for a long time. And so after UC Davis, Tyler went to MIT for a postdoc to work with Dr. Kristen Bergman, where he focused on Neoproterozoic carbonate sedimentology in Svalbard, and essentially applying what he learned from the modern microbial mats and carbonate systems in Antarctica to the ancient. And today he'll be sharing with us some of his research on microbial carbonates in icy environments. And so welcome, Tyler. Excited again to have you here talking to us today.
All right, thank you very much. I'm going to share my screen here so that I can get this started. And yeah, thank you for that introduction. I hope that everyone can see this all right here on Blue Jeans. But yes, as Francis mentioned, I am Tyler Mackey, and today I'll be really focusing in on microbial carbonates, and in particular, microbial carbonates of the cryosphere, so cold environments on Earth. And one example of those is what you can see on the side of the screen here, these beautiful charismatic decimeter scale columnar stromatolites that are growing at the bottom of Lake Joyce in the McMurdo Dry Valleys of Antarctica. And so this work is really coming out of my graduate work, as Francis mentioned, but drawing from some work that I've continued doing through my postdoc, and now I have a student carrying on here as a master's student at UNM. So I'm excited to share both some work that I've been working on for a while and some fresh new results from my master's students. So that should be a lot of fun. But I'd like to highlight first off that this is a collaborative venture. This work is not all coming out of me. This is coming out of a wide range of scientists who have a wide range of interests, from microbial ecologists with Megan and Dylan and Anna Youngblood here to limnologists with Ian Hawes. There's a wide range of expertise that go into an interdisciplinary geobiology study like what I'll be presenting here, including actually Justin Lawrence, who is on the call here. He came out to help with our field season back in 2014. So one of the reasons that I find these environments just so fascinating is that they give us a window into different time periods in Earth history when Earth may have been operating very differently. And so they're really complementary to some of our more traditional field sites for studying microbial carbonates. You might imagine going to the Bahamas and swimming around in Uwid shoals, or diving down to a stromatolite and lopping off a piece to take back to your beachside lab and explore there. And so at first blush, when you think about Antarctica and flying over this icy landscape, it may not seem quite as enticing as a field site to explore microbial carbonates. But there are environments here where I'd argue you can ask questions that you can't ask anywhere else. So there are a number of these meltwater oases in Antarctica. This is one of them that we'll be talking about today. This is perennially ice-covered lake with about 5 to 6 meters of ice on it year-round and tens of meters of liquid water below that. In these lakes, you have just beautiful microbial communities that form very complicated morphologies due to the interaction between the microbes that are living there and their environment. So in this image, what we see are these delicate webbed pinnacles that are a product of the activity of the microbes. And in some cases, these are mineralizing. This is calcium carbonate that is precipitating within these microbial mats, capturing that peaked and webbed morphology. And one of the questions that I'll be getting into today is what can we say about the activity of these communities and how they would be preserved in the rock record by looking at the carbonates that are associated with them. So working in these sorts of environments requires a different sort of infrastructure than you would have in the Bahamas, for example. We typically set up a field camp on the valley floors. We set up a dive camp out on the lake ice where we have a field lab and a dive hut. And then we have to get through the ice. So in this case, we're melting a hole with a metal coil that pumps heated antifreeze through on a closed circuit. And over the course of a couple of days, melts its way down through the ice cover. And once we're there, we can start to explore. So this is me getting ready to go into Lake Fryxell, which we'll be talking about later on today. Diving in with a dry suit and surface applied air. My collaborator, Peter Dorn, is going to hand me a camera here to take down and survey the microbial mat communities here. And so this gives a bit of an introduction to what the ice cover looks like and how it can modify the growth environments of these mats. So first off, we'll notice this is a thick ice cover. It's multiple meters thick. It has lots of bubbles in it that represent the incorporation of dissolved gases that are isolated from the atmosphere. You can see pods of sediment that are working their way down through the ice cover that will then fall to the lake bottom when they intersect cracks and form mounds on the surface. And then when we look at the lake bottoms, we're in these very still water depositional environments where there are no waves, no currents, no large-scale grazing animals to disrupt the way these microbial mats are growing. So we can explore their growth across basins and really interrogate how different environmental parameters are affecting the way that they grow. So this is a lot of work. We're going through and we are advancing slides, but it's a lot of work to get there and actually ask these questions. So why would we do this given that these are arguably weird environments in modern settings and may not be volumetrically significant in Earth history? I think that it really does give us an opportunity to ask questions that we just can't ask in more temperate carbonates. For one, we can use these modern lake deposits to better understand recent paleoenvironmental records. So thinking about paleolakes in Antarctica, where we have beautiful carbonates preserved through different changes in climate. So interpreting these are really important for reconstructing how the climate of Antarctica has changed through time, or say reconstructing the paleoenvironments where we might find similar facies on Mars, just as an aside. And then they also give us insights into pivotal periods in Earth history. So Snowball Earth events where these sorts of marginal facies on Earth today may have been much more widespread. And this is one of those projects that Francis alluded to that I really focused on in my postdoc. These are some really beautiful Snowball Earth lacustrine carbonates from Svalbard, where we have layers of microbial carbonates here with this beautiful texture, then coated in intervals of sand deposition before finally being buried under glacial diamictite. So by better having facies models to interpret these sorts of environments, we might be able to interrogate these really pivotal periods in Earth history. And finally, these lakes serve as really beautiful natural laboratories for exploring why microbial mats grow the way they grow. And here we're seeing a webbed pinnacle where it's a morphological analog for some really delicate filmy microbialites that are present in the Archean, even though the chemistry of the setting is completely different. So it gives us an opportunity to interrogate why these morphologies emerge where they do. So all these questions really motivate coming to Antarctica and digging into some of these freshwater oases. And we do a lot of work in the dry valleys of Antarctica because there are so many of these freshwater oases present that we can use them as natural laboratories. So we're pretty much due south of New Zealand at this point. And today I'll be focusing on Lake Joyce and Lake Fryxell as two case studies in this environment. Now, both of these lakes have multiple meters of ice on them year-round, but they have dramatically different lake histories and geochemistry that affect the way carbonates are precipitating in them. In Lake Joyce, we have a setting where we have oxygenation that extends down through the water column to depths where there is insufficient light for net photosynthesis. Whereas at Lake Fryxell, we have a setting where we have water column anoxia before we reach a depth where we have insufficient light for oxygenic photosynthesis. So in this case, we create oxygen oases where there's just a skin of oxygenation within the microbial mats with anoxic water above and anoxic sediments below. So these two examples really give us a lot of latitude to explore how and why microbial carbonates precipitate here. So the main questions that I want to approach today are really asking where and carbonates are precipitating in these lakes, and then how do these preserve evidence for microbial activity. So with that, jumping into Lake Joyce here to explore this well-oxygenated lake environment. And one of the first kind of foundations for this study is understanding that the stromatolites that we're looking at here are really relics of different lake conditions. They're currently not receiving enough light for net photosynthesis, but instead have been drowned by lake level rise to the point that they can no longer accumulate biomass and are now slowly decomposing in this water body. So what we see is a legacy of lake change and conditions through time. So when we look across the lake, even though these have been really slowly decomposing for decades, they are in a stably stratified water body that has maintained a consistent structure through that growth period. If we look at Lake Joyce water column here, this is the conductivity, so the salinity of that water body essentially. And we see a very dramatic structure in that water column where there's a freshwater lens on the top, the ice is above this here, and that freshwater lens increases with a little bump in salinity and then a much larger bump in salinity where the stromatolites are growing in this density gradient. And so this is showing us the history of the lake level in the salinity, where the meltwater is coming in and filling this freshwater lens, essentially expanding the lake above the stromatolites. So it's maintaining a very still water environment. So we can think about that schematically here as a history of where these stromatolites were growing, where we have a limit for net photosynthesis that at one point in time, the stromatolites that are now here around 20 meters depth were growing. They were in this density gradient that gets set up. Then we have a period of more meltwater influx that is essentially building this freshwater lens on top. And that limit for net photosynthesis follows the ice cover and eventually shuts down stromatolite growth. So when we look at these stromatolites, we're looking at a history of a changing environment. Another thing to assess here is the effect of the ice cover. And this ice cover really dramatically contributes to the carbonate chemistry of the lake. If we look at the pH, it's fantastic when you can scale your conductivity and your pH on the same axis. The shallow waters are up around pH of 10. But if you were to remove that ice cover and equilibrate with the atmosphere, it would go to around a pH of 8. So this ice cover dramatically affects the carbonate chemistry by preventing equilibration with atmospheric CO2. And so when we think about the carbonate environments here, we're not necessarily looking at something that's in equilibrium with the atmosphere. And then dissolved oxygen is also trapped by this ice cover. So we have dissolved oxygen here in milligrams per liter, well above atmospheric saturation. And we can see that this oxygenation extends well into the stratified water body of Lake Joyce, just starting to decrease once you reach these much stronger stratified areas. And so in the shallow parts of Lake Joyce, this gas supersaturation leads to bubbles nucleating around and within the microbial mats, here causing them to be buoyantly disrupted by the gas bubbles. There's a gas bubble at the peak of this finger sticking up from the lake bottom. And so it's a very dynamic environment in the shallow water where the mats are being disrupted by gas bubbles nucleating in and around them. The cold water and high oxygen contents, though, really do make this a well-oxygenated environment year-round. Even though these lakes have 6 months of winter darkness, work in an adjacent lake, Lake Hoare, that has very similar water chemistry, has oxygenation that's pervasive down to here, like 5 centimeters depth over the duration of the winter, just because of these extremely high levels of oxygenation and low levels of respiration because of the cold water. So all this comes together to give us a really interesting and informative environment for carbonate precipitation and understanding how the ecosystem can enter into the rock record. So I mentioned earlier that these stromatolites receive insufficient light for net biomass accumulation. They're now right around here, or they were when I looked at them in 2014. And so they are now down at this depth where they receive insufficient light for photosynthesis, but they're still within the zone of being well oxygenated. So we don't expect that these would be anoxic at any point during their growth or degradation. But if we look inside of these are truly stromatolites. They are layered growths of microbial communities. Here we have an X-ray CT scan of one of these stromatolites showing the opaque white as the layers of carbonate that are accumulated within the microbial mat. And the blue and red here are organic biomass and mud sediment. So this is just a line drawing schematic showing these zones of layers to kind of guide our eyes. And what's really fantastic about these is if you look at the innermost layers of these stromatolites, we can see bubble molds that are similar to the morphology of what we see growing and just being disrupted in the shallowest parts of the lake today. So when we think about these ancient stromatolites, what we really should be thinking about is one of those buoyantly supported pinnacles in a shallow water environment, having carbonates precipitate on it, and then serving as a template for successive layers of stromatolite growth. So it's definitely not consistent with the environment that these are in today. But this evidence gets preserved because of the carbonates that are precipitating here. So at the peaks of these stromatolites, we see molds of bubbles that are surrounded by carbonates. So these spherical molds come from the crest of one of these disrupted stromatolites. And these bubble molds really indicate that mineralization was early. So it had to mineralize while the bubble was still present in the microbial mat. But if we look at it under SEM, which is what the image is here, we can also say that precipitation didn't really continue after this time. So zooming in on this surface, this is the inner surface of a bubble mold, we can see that there are imprints of cyanobacterial filaments that are still maintained on the surface of the bubble mold. So if we think about a mechanism for this, we have a bubble captured by a film of cyanobacteria. We have carbonates growing away from that surface, and then we lose the bubble, and we don't have carbonates filling in that space over time. Instead, we just have outward growth of those carbonates. So it really helps us localize where this carbonate was precipitating. We can also situate this carbonate within the mats. So how close the mat-water interface are we through some of the textural data of the carbonates. Again, if we look at the crest of one of these peaks, we have this little tube that's at the crest, and then the calcite actually forms these really beautiful kind of hemispherical botryoids, they're called, where they're growing crystals that form a fan on the sides of the stromatolite. And what's particularly exciting about these is that the sediment that is incorporated into them, so from settling out from the water column, sediment that's been brought in by meltwater, we see that the surface of this botryoid has abundant sediment on it, whereas the side and the underhang do not. And so we're looking at a setting where this carbonate was precipitating sufficiently close to the mat-water interface that it could actually shelter the lower parts of the microbial carbonates from incorporation of sediments. So these are really precipitating right at the mat-water interface. So if we return to our questions of like, what are the where and when are the carbonates precipitating? We've kind of gotten that with the bubble molds and the surface relief. But if we want to know what sorts of microbial influence is here, we need to think about the physiology of the mats that would be present at that mat-water interface. And one of the first things that we think about in this setting is really photosynthesis. This is a photosynthetic cyanobacterial mat. And so the metabolism that we expect to be co-located with carbonate precipitation is that photosynthetic activity. So to see how photosynthesis may be affecting that carbonate precipitation, we do things like microelectrode profiles, going down by hundreds of microns at a time into the mat and pulling out the, in this case, pH that is within the microbial mats. And so here we can see that from the water column, where we already have this hugely elevated pH level, into the microbial mat, we have an increase above pK2. So we're going from having primarily bicarbonate to having primarily carbonate in solution, which would dramatically affect our carbonate saturation state. We can tie this to photosynthesis by actually looking at how this changes with expected irradiance. It would have been great to have this with actual irradiance, but our light meter broke, so we modeled it. But even with that caveat, it actually follows this really beautiful diurnal cycle of pH versus irradiance with a lag. And I don't think we can say from this data whether that lag is due to the rates of diffusion or due to limitations of our model for irradiance. But I can say that here we have clouds that came over on that day, so we can actually tie it to what's expected to change in the light levels. And so this sort of cycle of pH modification in the mats really highlights how strongly the microbial photosynthesis could be affecting our carbonate chemistry. If we wanted to track this in a modern setting, we could do things like taking pulse amplitude modulated imaging to co-locate rates of photosynthesis with different textures on the mats. You're just grabbing an image of ridges from one of the Lake Joyce microbial mats that we examined, and these PAM images showing where we have higher rates of photosynthesis on the ridges. But since we're looking at recent stromatolites that are no longer photosynthetically active, we have to infer potential morphology or activity. And one of the tools that we have for this is the morphology of the microbial mats. If we remember that these innermost parts of the stromatolites, this is a cross-section through the innermost growth of one of those stromatolites. What we're looking at here really is one of these disrupted layers of microbial mat that's been torn off the lake bottom by a bubble and then successively coated with carbonate. So we might be able to say, well, there's more light on the sides than there is on the sheltered part, so maybe we can track the rates of photosynthesis with where we are on these structures. That's fairly limited because we have other factors that could influence this, like the diffusion of ions through the mat. But it does give us sort of a a context for the way that these are growing. But these stromatolites have multiple layers. And so we can look at how these carbonates are precipitating through time as this lake is changing and light levels are decreasing, and really try to interrogate how the microbial activity of these photosynthetic mats affects carbonate precipitation and can leave a signature there, since this environment is changing so dramatically. So to do this, one of the tools that I focused on is the isotopic composition of these carbonates. And when we're looking at the delta 13C value, so the carbon isotope composition, we can think about our different pools potentially having different compositions that we can track through time. We start out by thinking about our dissolved inorganic carbon, so our DIC reservoir, which here I've just put at 0. And we have a typical photosynthetic biomass offset where our biomass preferentially takes up the lighter carbon, and so it is fractionated relative to the bulk inorganic carbon pool. If we look at the water column of Lake Joyce and the delta 13C values of that water column, we see from our measurements of meltwater streams coming in right around 0 to -5. We have shallow water areas with a clear signal of net photosynthesis where we have very positive delta 13C values, and deeper areas where we return to net respiration and have those values becoming more negative, comparable to or more negative than the meltwater streams that are coming into the lakes. So our stromatolites are within this context of metabolisms structuring our dissolved inorganic carbon pools. And the reason that we can get this really dramatic expression is that our ice cover, again, is preventing mixing and equilibration with the atmosphere. So we're really in a semi-closed system where our metabolisms can fractionate the carbonate pool that they're in. And so to preserve this, we can look at calcites. Calcites are fractionated relative to the dissolved inorganic carbon pool that they precipitate from by a relatively small amount that is predictable. And if we think about having photosynthesis taking place alongside carbonate precipitation, we can start to get some really interesting relationships forming in the pore waters of these microbial mats. So we have, let's say, our typical photosynthetic biomass accumulating this inorganic carbon. Then we can imagine that the pore water geochemistry can also shift to more enriched values, higher delta 13C values, when it's co-associated with photosynthesis. So we really have two levers in this lake. We have what's happening in the overall water column, so we can evolve this partially closed system, but we can also evolve what's happening in the pore waters of the microbial mats where carbonate is precipitating. And so when we look at these records of stromatolites, these are the two levers that I want us to have in mind to try and tease apart. So here are some isotopic data from these stromatolites. This is a compilation of 7 different stromatolites, and here we have on the x-axis a distance from the center of the stromatolite. So we're looking at something here over just over a centimeter of radius of one of these stromatolites. And they're lumped by zones based on the petrography of the carbonates. So those relationships of how the stromatolites were growing and how they changed through time. On the y-axis, we have our delta 13C values, where we're looking at those carbon isotopic pools. And what is sort of a first order view here is things become more enriched through time. We have our innermost zone to our outermost zone, the mean changes, but it also becomes much more consistent. We go from having essentially a 6 per mil spread in delta 13C values to a much tighter collection of delta 13C values. So what we're really tracking in this is a combination of these two levers. We have our delta 13C values really highlighting this evolution of water body chemistry in a partially closed system with net photosynthesis, where through time, this inflowing meltwater is being fractionated by net photosynthesis in a very stable stratified water body. If we think about the microbial influence, so what's happening in that pore water chemistry, that's where we can go back to our data sets and look at the fact that these are becoming much more consistent. So we are losing a lot of that variability from local photosynthetic effects as this stromatolite grows. So we're going from essentially having huge pore water effects, fractionating the delta 13C values where the carbonate is precipitating, to having very small effects where the carbonate is precipitating. And this is a beautiful signature of changing microbial activities through time. But I think that it also poses a challenging question to us, is that there aren't always carbonates precipitating here. So what do the carbonates themselves mean about microbial activity? We have large gaps in where and when carbonates are precipitating, as well as how they're precipitating. If we look at the, if I can get my slides to advance here. Fortunate. Let's go ahead and talk to you about this then while my computer thinks about life. In the innermost parts of the stromatolites, we have the same petrographic textures as we have on the outermost parts of the stromatolite. So these are those really beautiful acicular botryoids that are present both on the inside and the outside, where we expect to have the most different types of environments and microbial activity taking place. So what this suggests is that our microbial activity itself isn't the only parameter that's driving where and how our carbonates are precipitating. They are precipitating the same way when they have the most light as when they have the least light. So it's largely modulated by some external environmental control, giving us the carbonate where we have it. And since I'm still not able to advance my slides, I will stop sharing quickly and start that back up. All right, clearly advanced a number of slides there. Cool. So here are those inner and outer layers I was talking about, where our shared textures are present, where we have the most photosynthetic activity and the least photosynthetic activity. So coming back to our driving questions here of where and when carbonates are precipitating. And here we can see that there is early precipitation right near the matte water interface, referenced from those bubble molds and the surface relief of the carbonates. And then these preserve evidence for microbial activity by showing us local photosynthetic effects. And these do decline with lake level rise, at the same time that we have the largest net photosynthetic effect of modification of that pore water, of that water column dissolved in organic carbon composition. And this is all happening in a context of lake level rise of around 0.2 meters per year over this course of decades. So through time, these photosynthetic signatures really change in the stromatolites that we see here. So we've looked now at Lake Joyce, where we have these well-oxygenated microbial mats with varying levels of light for photosynthesis. Now we're going to go to Lake Fryxell, where we have anoxic waters and still have enough light for photosynthesis in these more heterogeneous chemical environments. And so here we have a water column view of Lake Fryxell, where we have our dissolved oxygen here increasing to, again, extremely high levels, well above saturation, and then crashing out down to essentially zero within just a meter or two in the water column. And this is, again, our conductivity, so a measure of our stratification. If we zoom into one spot on this lake, again, here we have a record of slower lake level rise at around 6 centimeters per year instead of the 20 centimeters per year that we had Joyce. But if we zoom in on one part of this water body, we can see the change in oxygen levels here as our surface irradiance decreases as well. But the light decreases less rapidly than our oxygen, and we can do net photosynthesis down to around 0.1% of the light that's reaching the top of the ice cover. So these are very shade adapted communities. And if we look at what's happening within the microbial mats in these different areas, in the shallow parts of the lake, we have very high ambient oxygen levels, which increase into the mat and then decrease. As we look down to the anoxic parts of the lake, we have much lower levels of oxygenation, these like just 1 to 2 milligrams per liter here, but anoxic water above and anoxic sediments below. So we have photosynthesis taking place in these very diverse chemical environments. If we look at the different metabolisms that are present within these mats, this is work that Megan Dillon did looking at metagenomics of the mats that we collected here from the different depths, and then from the top of the mat, the middle of the mat, and the bottom of the mat. So kind of arbitrarily based on the structure of the mats, selecting these bins. What we see is that the metabolisms change quite dramatically in these different oxygenated microenvironments of the lake. And we have things like sulfate reduction becoming much more abundant as we go to greater depths in the lake. So if we want to explore the way that metabolisms are preserved in carbonates, this sort of context should really give us pause. In Lake Joyce, we had a very simple system where it was well oxygenated year-round and through changing light levels. Here we have a lot more in play, where over here just less than a meter in water depth, we have dramatic changes in our oxygen composition, in what metabolisms are present, and at least episodic anoxia in these oxygen oases. So through all of these different ranges in Lake Fryxell, where we have so many different metabolisms present, so many different geochemical conditions, we still have carbonates precipitating and potentially capturing ecological information. So as we go down through this water column, the carbonates are in environments that range from being hyperoxic to being euxinic with free sulfide present. And so our questions here remain: where and when did the carbonates precipitate? And what metabolisms were they associated with? How are the microbial communities being preserved in these environments? So if we want to understand this record of metabolism in microbial communities with carbonate precipitation, we can start by just looking at the mats that host these carbonates. So this is actually the view that we saw in the video from a different vantage point this time. This is the camera that went down through the ice cover with me. Here's Peter handing it off. And what we can do is swim around and essentially map the way that the mats are growing on the lake bottom. This is a conductivity depth and temperature measuring instrument. And these are two lasers 3 centimeters apart per scale that also serves as an altimeter to show how far off the lake bottom I'm swimming. So with those data sets, we can map where different mat compositions are found relative to the geochemical transitions in the lake. And what we find is that this oxycline, where we go from having in the shallower parts hyperoxic to suboxic into this oxygen oasis environment where we just have seasonal oxygenation within the microbial mats, that really specific microenvironment is present in these green flat mats. And this is found over the span of like 20 to 30 centimeters of water depth. So really at the resolution of our mapping across the lake bottom here. And if we look at the mats themselves, they can tell us about how this oxycline has been shifting through time. There are the shallower water honeycomb here, our deeper water mats where we start to lose the beautiful pigmentation. If we look at the a core through one of these mats right at the transition between these two mat facies, we see is that this flat mat is growing over honeycomb mat. So showing that this community associated with these oxygen oases is growing over the honeycomb mat that is associated with more oxygenated lake environments. And as we go to the deeper end of these green flat mats, we see a number of layers of these microbial mats, all on top of what are now heavily degraded honeycomb structures. So we're looking at really a relic of decades of shifts in the oxycline here. So this is all well and good, but if we want to understand how the carbonates fit into this, we need to look at where the carbonates are distributed within this history of changing lake environments. And so one way we can do that is by taking cores through the sediments and tracking how the carbonates interact with these microbial mats and where they're preserved. These are two sand mounds that are present on the bottom of Lake Fryxell from those episodic pods of sand that fall through the ice cover onto the lake floor. And what we can see from these cores that I collected here is that on one sand mound, the carbonate is present at the top of the course. This is from work that Francis has published on the sedimentation here and the patterns of sediments in ice-covered lakes. In our other mound, we see that the carbonates are present exclusively at the bottoms of the cores and not in the mats that are on top of it. And these are only 1.5 meters apart from each other across the lake bottom. And so what this tells us is that the carbonates that we see in Lake Fryxell are episodic in nature. They're not precipitating all the time in these mats. So we need to take their distribution with a grain of salt when we're trying to interpret how metabolisms are preserved. They may not be showing us the current metabolic activity of the communities that are there because they are from different times in the evolution of this lake. This is work that my current master's student, Jared Clance, is digging into, looking at how these carbonates are precipitating and what sorts of evidence there might be for metabolisms preserved in the carbonates themselves. And one of the tools we found is really useful for this is cathodoluminescence, which uses electron bombardment of the carbonates and the luminescence of the carbonates themselves to track how much manganese and iron is incorporated into the carbonates. So if we look at our just EH-pH diagram here and predictions for where different observations come through, as we head to more oxidized conditions, we go from having abundant dissolved iron in solution incorporated into carbonates, dissolved manganese, to having areas with more abundant manganese, less iron. And as we go through these transitions, we go from having non-luminescence to dull luminescence and then into brighter luminescence. So it's a qualitative record of how redox may be preserved in these carbonates. So I'm going to show two samples, one from well-oxygenated waters here above the oxycline and one from currently oxygen oases environments where we have just seasonal little whiffs of oxygen within the microbial mats. And what we see in this is that there is zonation within the carbonates consistent with differences in manganese and iron composition, so these redox-sensitive ions incorporated into the carbonates. And that varies with depth. So it means that we are still tracking with changes in an oxycline when these carbonates were precipitated, similar to really what we see today. This is fresh data from Jared as of Wednesday. So he's paired these redox proxies from cathodoluminescence with maps of the composition in microprobe transects. So it's showing the manganese and iron in ppm here going through these zonations in the carbonate. And so we see is that this zonation really is consistent with fluctuations in pore water redox through time, giving us different compositions of our carbonates and the distribution of these manganese and iron redox sensitive elements. So the textures of these zonations and these carbonates within the mats also give us some information about mechanisms of precipitation. Here we have one of those really beautiful calcite botryoids again with a layer that is accumulated within the growth of the botryoid, showing growth banding. And if we pair this with cathodoluminescence imaging, we see that there are these series of bands in composition of our redox-sensitive elements that are both within this beautiful botryoid as well as growing through the microbial mat adjacent to it. And so this relationship of having the same patterns of redox-sensitive elements in a pore space where we have clean carbonate growing and in a microbial mat where it's growing through biomass really show us that these mats are passively being encoded or entombed within carbonates rather than actively driving the carbonate precipitation at the mat-water interface. So it's a completely different system to what we saw at Lake Joyce, where our carbonates were so intimately associated with photosynthetic activity. So here we have really passive mineralization of the mats. So if we want to return to our questions here of where and when carbonates are precipitating, we can say that it's episodic from our evidence in the sand mounds and from our textural evidence of these botryoids entombing the mats. We can say that this carbonate precipitation is apparently within the subsurface of the microbial mat. And now the question that is really motivating the work is how these preserve evidence for microbial activity. Well, we know that these aren't capturing that mat-water interface environment where we expect there to be, say, abundant photosynthesis. Instead, they are tracking bulk changes in redox. So that zonation itself might still give us information about how the ecosystem is functioning at a more coarse scale through the time period that these are being mineralized. And so this is an area I'm really excited to continue digging into with Jared to really interrogate how the carbonates preserve annual, decadal, longer timescale changes in redox as the carbonates are precipitating within these microbial mats, and what that says about how our stratification may have changed through time in this ice-covered lake. We have these two very different environments here where we have oxygenation extending to depths where there's no longer enough light for photosynthesis. The carbonates precipitated at the mat-water interface. It was a beautiful photosynthetic signature. Then we have our Lake Fryxell setting where it's anoxic depths with sufficient light for photosynthesis, extending down into anoxic water bodies, and where our carbonates are passively growing within the microbial mats, capturing bulk changes in redox as opposed to characteristic photosynthetic signatures. So what lessons do these give us for really interpreting our paleoenvironments of these sorts of cold water carbonates? The first lesson I want to pull away from this is just how different these environments can be from each other. We had Lake Joyce here, Lake Fryxell here, both perennially ice-covered lakes, both essentially in the same valley network, but with completely different geochemical signatures and relationships between their microbial communities and their ambient water bodies, largely because of the history of the lakes, one rising more rapidly and one rising more slowly, really giving us a different evolution of the lake environment. We also have our atmospheric isolation that can amplify the heterogeneity within a single lake as well. So in Lake Fryxell, we see that going from hyperoxic to anoxic within a meter of the water body. So that's intensely stratified chemically and very stable through time. And I also want to highlight just that these changes in ambient water chemistry that we can have with a perennial ice cover really can be an alias for microbial activity changing through time. If we were to naively look at this as things becoming more enriched through time, indicating higher rates of photosynthesis, we would have the opposite interpretation of what we observe in the system, where our earliest signals are of the most photosynthesis with the most variable isotopic compositions. And then through time, with net photosynthesis at lower rates, we evolve to this more consistent, enriched delta 13C value. So picking apart that environmental change from local pore water chemical changes is really important when we're interpreting these sorts of depositional environments, and could give us a completely errant interpretation of these sorts of ancient settings. And finally, even though there are a lot of caveats in these settings, they really have a very rich record of microbial activity if we know how to read it, and we can pull apart the context of those changing environments with the carbonate precipitation, timing, and location. So when we expand our views and look into these sorts of changes in environment through time and changes in potential microbial activity, I think we can say a lot about ecosystem function and what might be preserved and be interpretable from the rock record or microbial carbonates that we would hope to find elsewhere. So thank you, and happy to take any questions here.
I'll give you a little applause. Thank you, Tyler.